Okay, hi. So in today's video, we're going to be explaining about biology, meiosis phase, and then we go from prophase, metaphase, anaphase, and all of that. And today I'll be using this to be a model of mine for today's explanation. And then right here you can see the homologous chromosomes right here. There are three pairs of them, and these are all attachable, means I can stick back on to explaining further on. And then here are the centrosomes, and then the microtubules, and right here this is the prophase number 1. And then later on I'll be drawing different circles since the chromosomes are going to be moving around throughout the phases. So this is how it's going to be looking like for now. And yeah, so this is how it looks right now, but then later on we're gonna be experiencing synapses, means that the mother chromosomes and the father chromosomes, the genes, are gonna be really close to each other later on. They're gonna be kind of very close to a point where they are crossing over. There are different assortments in genes, such as the law of segregation and law of the independent assortment, and this is how they look like for each of them. So the difference between law of segregation and independent assortment, you can tell that the law of segregation is more normal, it's very basic and all that. But then for the independent assortment, you can tell that there's a little bit of the mix and match going on here at the bottom, and there's like red and blue, blue and red, but then compared to law segregation, it's just red and red, blue and blue. And these assortment, these law segregation or independent assortment will be happening at the anaphase. But I'm just telling you here to have a better idea of what's going to be happening, so later on you'll have more idea of why the assortment happens and why is it like that, and the different kinds of assortments will lead to different kind of results. And it will look something like this, and where their legs are crossed over, and I hope you can see it properly. Let me just attach a picture right here for a better quality picture to show you guys, because their chromatins met and now they exchanged their legs and genes. Now that it looks like this. Over here we're still on prophase 1. Later on we're going to see some different changes. We're going to be moving on to metaphase 1. This is the metaphase 1, and after the prophase, you can see that the chromosomes have moved to the each polar sides right here, transformation right here, and then the chromosomes will start to be at the center where the microtubules are starting to attach to them, and then later on they will start to pull them apart, which will happen in anaphase. And the anaphase will look something like this at the end, where the chromosomes are now split up. They were together and they split up in anaphase 1. It'll look like this, and where the microtubules are kind of like pulling them closer to them, like that. So this is how the telophase 1 would look like. And they split them into 2, and there are 3 sister chromosomes right here. And right here you can tell that they are separated into 2 parts. Any prophase 2 will be looking something a little bit like this, or you could be separating them. And right here you can tell that the sister chromatins are now separated into 2 parts, where they are forming two different individuals. And right here you can tell that the microtubules are now pulling them farther. That's going to be happening in the metaphase, but right here you can see that the whole thing is happening all over again, but then except that the genes are different now. So this is how the anaphase 2 would look like, and you can tell that like the microtubules has already pulled them. Just now it was connected, but now they pulled them apart, which created something like this. And after the anaphase 2 is done, we're going to be moving on to telophase 2, which means that these are going to be pulled apart like that, and they'll be creating like that. They'll be pulled apart, creating 4 parts, which is the result. This is the 4 daughter cells, which is the unduplicated chromosomes together but separated in 4 parts. And then the 4 daughter cells are different from each other, and because they're the patterns that they have are different too. And then the anaphase that we just experienced over here breaks down the proteins and holding the sister chromatids together, and then it allows them to split and then to this.